Thank you very much. Good afternoon, everybody. I know it's been a long day and an exciting day. I was kind of switching gears a little bit, and just want to talk to you about the work we've been doing on the endometrium. So the outline of my talk is, what I'll do is try to divide my talk into three parts. Talk to you about mechanisms by which we think HCG regulates uterine receptivity, and then just share with you some of the work we've been doing in trying to understand mechanisms by which endometriosis, which is the disease I work on, impacts the uterine receptivity, and also the effect of HCG on the endometrial response as far as it relates to the human situation, and some of the work we've been doing to try to correct endometrial receptivity, as you've heard, some of the issues that we have with assisted reproductive therapies. So in terms of implantation, the window of receptivity is very, very critical. When we think about the window of implantation in all species, there's a very narrow window of time during which both the blastocysts and the uterus are synchronized, and at that time, that's the most optimum time for implantation to occur. Now, if implantation occurs outside of that window, as this diagram from Wilcox that was published quite some years ago, there's a huge increase in the miscarriage rate, suggesting that anytime you're outside of that window, you're not going to get a successful pregnancy. The other issue we have is that in the human situation particularly, we have an enormous loss with regards to pregnancy and implantation. Up to about 60% of the pregnancies are lost very, very, very early. And this is very unusual compared to other species that have a much, much more effective way of implanting and are much more successful. So the human has a tremendous wastage when it comes to the implantation process. So the question has always been, why is it that it's different as far as the human is concerned? And that's what my lab has been addressing for many years. And the thing we've been really interested in is whether, like in other species where the embryonic signal has such a profound impact on modulating the endometrium, does the same thing happen as far as women are concerned? And of course, the hormone of pregnancy, as far as the human is concerned, is chorionic gonadotrophin. And in other species, particularly domestic species, the embryonic signals really modulate the response to the endometrium. So what we've really asked is whether chorionic gonadotrophin, in addition to its major role in rescuing the corpus luteum and maintaining progesterone production, whether HCG itself has an impact on the endometrium. And many years ago, work from C.V. Rao and other groups had shown that in endometrial tissues, there are the receptors for the chorionic gonadotrophin receptor in the endometrium, and that it does play a role in vitro with regards to regulating the decidulization process and also regulating some of the other responses in endometrial cells in vitro. Now, the issue, of course, is that how do you do these sorts of experiments in the human, because it's almost impossible to try to mimic what we do in other species, particularly with domestic species. So several years ago, we decided to look for a surrogate model that we could potentially use to try to understand embryonic signaling and implantation processes as it relates to the human situation. So my lab for many years has worked on using the BABOON model, and the BABOON is a very, very useful model. It's an old world primate that has the cyclicity, which is very, very similar to the human. You can very easily monitor the cycle because of their responsiveness to hormones, and you can simply look at the sex skin and tell whether they're in the proliferative phase or the secretory phase. And they only have a single placenta, unlike the rhesus macaque, which has a double placenta. So many of the features that you see in early implantation are very, very similar to what you find in women. So it's an ideal model, besides the fact that it's not endangered. So it's a human primate, non-human primate that you can use extensively. And what we've done is use this model to ask specific questions as it relates to the implantation process. So in my mind, we think about the implantation process in 3 different phases. The initial phase is one that we are probably most familiar with, is when estrogen and progesterone during the normal menstrual cycle set up the endometrium to allow it to develop and appropriately respond to the presence of an embryo. And then the second phase is during that window of uterine receptivity, when the embryo and the endometrium are synchronized, and the signals from the embryo are initiating responses to the endometrium to allow for the early apposition and attachment that then have to precede this third phase, which is where there's invasion into the endometrium and the establishment of placentation and the interaction with the spiral arteries. Now, obviously, with phase 1, you can study this in women. There's no problem. You can get endometrial biopsies, but for the most part, anytime you get an endometrial biopsy from women, it is also in a situation where there is a pathological need to do an endometrial biopsy. It's very difficult to get normal endometrium from women. But to study phase 2 and phase 3 in vivo, obviously for various ethical and moral reasons, you're not going to be able to do that as far as women are concerned. So based on that, we've developed the BABOON model for several years to try to understand what happens during these very critical phases of endometrial responsiveness to see to try to study the mechanisms that contribute to successful implantation. So the question we've asked is really what happens once the endometrium, and you heard some of that work earlier in the last session about the development of the endometrium, the very dynamic changes that this tissue undergoes, the remarkable responses that happens every single cycle, which the secretory phase, which is governed by the increase in progesterone that sets it up to allow for the endometrium to respond to the developing embryo. And we simply ask what happens now if we superimpose the embryonic signal upon this receptive endometrium and see what the endometrium does to respond to the presence of an embryo. Now, the non-human primate, the baboon and rhesus macaques, are just as inefficient as the human female in terms of implantation. So we had to come up with a model system that mimics the endocrine milieu within the uterine environment that is similar to what happens with the presence of a blastocyst to try to understand what the signaling mechanisms are. And we set up what we call the simulated model of pregnancy in the non-human primate. And what we do in this model system is that we cannulate the oviducts of these animals and exteriorize a very tiny polyvinyl cannula to the exterior under the skin. So we can go in at any time and simply expose the polyvinyl cannula and put in an all-Z mini pump that contains chorionic gonadotrophin or any other component that we want to test, and infuse in a very programmatic manner into the uterus to try to mimic the endocrine milieu, as I said, within the uterine environment. So our model here is we take normally cycling animals that we can monitor constantly by looking at the blood samples to make sure that when they ovulate, and then on 6 days after ovulation, we begin the infusion of chorionic gonadotrophin for the 4 days, which is the 4 days during which the blastocyst is free-floating within the endometrium. And so this is also the time that the blastocyst first begins to produce chorionic gonadotrophin. It is the window of time that the corpus luteum becomes most responsive to chorionic gonadotrophin. And it is also that window of time, like I said, that the blastocyst is free-floating before it begins its implantation in this species around day 10, about a day or two later than the human situation. And when we then took endometrium after we had done this, what you find is there's a remarkable response to every single cell type within the endometrium. The luminal epithelium undergoes this classic plaque response where there is increased proliferation of the luminal epithelium, and it's confined to the luminal epithelium, and there is sensitization of the luminal epithelium that opens up with increasing glycogen biosynthesis plus the opening up the tight junctions to allow for the trophoblast to migrate between these cell lines. The underneath of the luminal epithelium, the stromal fibroblasts undergo a myofibroblast transformation, and they're characterized by the expression of alpha-smooth muscle actin, and the glandular epithelium becomes highly secretory, and a large number of secretions that are important, that are progesterone-regulated and HCG enhanced, that are important for the implantation process, are initiated in response to chorionic gonadotrophin. So the question then was, we've been able to show that these responses were happening in the non-human primate. We've also wanted to know whether there was a responsiveness with regards to the rescue of the endometrium as far as women were concerned. And in this particular experiment, we were able to get endometrial biopsies at day 23, which is putatively just outside the window of receptivity, or treat women with either 200 milligrams of intravaginal progesterone suppositories between days 18 and 26, or a single injection of chorionic gonadotrophin on day 19. And what you can see here is when you did the endometrial biopsies on day 26 after either HCG or progesterone treatment, you inhibited the normal apoptotic cascade that you see during the later stages of the menstrual cycle, suggesting that even the addition of chorionic gonadotrophin or the increase in progesterone was preventing the normal apoptotic response that would occur. And we confirmed that by essentially looking at tunnel labeling and tunnel staining, and you can see that in the absence of treatment, there is very intense tunnel staining of the endometrium on day 26, but in the presence of chorionic gonadotrophin or progesterone, you see a remarkable decrease in the amount of apoptotic signals based on tunnel staining with regards to the endometrium, showing that the endometrium has been rescued from undergoing the normal apoptotic process that occurs later in the menstrual stage. We also see that we see the increase in progesterone levels in response to the intravaginal progesterone treatment, and this is the H score for tunnel staining, which is significantly downregulated in the presence of HCG. So we're getting these responses in the non-human primate, creating an environment that is similar to what's happening during implantation. So our next question was, what are the signaling mechanisms that are associated and initiated by the presence of chorionic gonadotrophin within the endometrium? And what does the embryo do at the time of apposition to alter the endometrial environment in response to the embryonic signal? The one thing we looked at the normal classic pathway of signaling with HCG binding to its receptor and activating the cyclic ANP protein kinase A cascade. And what we found was that in the endometrium, even though this is the response that you see in the corpus luteum, this response is completely attenuated as far as the endometrium is concerned. Instead, the endometrium has adapted a process very similar to what the beta-adrenergic receptor does, in that when chorionic gonadotrophin binds to the same LHCG receptor that's on the endometrium, it activates a ERK kinase pathway, essentially activates the MAP kinase pathway that allows for translocation of ERK into the nucleus and essentially activates ELK1 to induce a whole series of endometrial changes that we looked by microarray. And if you then used siRNA against the LHCG receptor, we were able to block this response. So the endometrium has adapted a unique responsiveness to the presence of chorionic gonadotrophin, and it makes total sense when you think about the endometrium that's sitting there bathed in large amounts of chorionic gonadotrophin, and one of the classic responses is desensitization of the LHCG receptor in the presence of this ligand, that if the endometrium is going to continuously respond during the time frame of implantation, when there's large amounts of this hormone being secreted, there has to be a modification of the signaling response to allow for that signal to continue to initiate the responses it needs to initiate without being desensitized like it does in the corpus luteum. So essentially, this is the mechanism by which the chorionic gonadotrophin is signaling within the endometrium independent of the cyclic ANP pathway. And with our microarray analysis that we did in response to this, we came up with a whole series of genes that were altered, and the ones we focused on primarily were the ones that we thought that had a significant functional response. So the glandular epithelium, in addition to responding to the chorionic gonadotrophin, increase two of the classic genes that most of you are aware of. It increases glycodelin and increases LIF, which are both critical for implantation, particularly LIF, because in the absence of LIF, there's no implantation. The stromal fibroblasts respond by changing its phenotype, becoming a myofibroblast phenotype, which is absolutely essential to transform these cells into the decidual cell, and also increases the expression of Notch1 that I'll talk to you about. And the uterine vasculature is altered by the increase in eNOS and iNOS, along with an increase in VEGF, to enhance the angiogenesis and the vascular development and the vascular remodeling that's important for the trophoblasts to migrate and attach into the spiral arteries. And each of these have very significant important functions in that it needs to modulate the immune system to maintain the endometrium and maintain the fetal allograft, to prevent the apoptosis, as I showed you, and initiate the decidualization process and enhance the vascular development. And we've actually focused on the role of Notch1 that we think is an important initiator for the decidualization response, and in the absence of Notch1 or the decrease in Notch1, the decidualization process is inhibited. So given the normal responses that we were seeing with chorionic gonadotrophin, one of the questions we had was what happens in a situation where infertility is a major problem with a disease like endometriosis. So again, with endometriosis, we've developed the non-human primate as a model, as a surrogate model. And because, as you well know, with endometriosis in women, it's diagnosed 8 to 10 years later. We have no way of understanding the early etiology of the disease. And what we do is, since baboons also get spontaneous endometriosis, but more importantly, we can induce the disease using Sampson's hypothesis, where we inoculate these animals with autologous menstrual tissue on 2 consecutive menstrual cycles, and they will then get disease for life. They have predominantly peritoneal disease, and as you can see, here's a laparoscopic evaluation 3 months after we've inoculated these animals with endometriosis, and the tissue looks exactly what you would expect with endometriosis, glands and stroma, and it's very, very similar to what you would see in the human female. And if you now look to see what happens globally with regards to the endometrial response, so this is an HCG-treated animal with endometriosis compared to animals without, and you can see that each of these responses, the luminal epithelial plaque response, the underlying stromal response, and the secretory response of the endometrial glands, are completely suppressed in the presence of endometriosis. Endometriosis, as you know, has progesterone resistance, and the interaction between the progesterone resistance and the ability of the embryonic signal to stimulate this response is completely affected in the presence of this disease. In addition, what we also found that the induction of endometriosis alters the overall endometrial global transcripts and results in the abnormalities decidualization, as well as an altered immune system, all of which are important in terms of implantation and are classic components that are associated with endometriosis. So having seen these global changes, our next question was, how does this then contribute to what's known about this disease and the abnormalities of decidualization? So as I said, Notch had showed up in our array, and Notch is a highly conserved evolutionary gene that has important roles in preventing apoptosis and enhancing both proliferation and differentiation. And if you then look at the expression of Notch at the same time that you treat the animals with chorionic gonadotrophin, you can see that there's an enormous increase in Notch immunostaining in the stromal fibroblasts that co-localize with alpha-smooth muscle actin, and alpha-smooth muscle actin is a transcriptional regulator of Notch1. So you're seeing a very, very early response that's important to bring about this differentiation. And looking to see how important Notch is for decidualization, you can use either dominant negative mastermind, which is a co-regulator with RBPJ to initiate Notch signaling, or use siRNA to block Notch signaling, and using each of these and then taking these stromal fibroblasts and incubating them under the conditions in which you induce destivalization, that is estrogen, progesterone, dibutyl cyclic ANP, you can see that when Notch signaling is completely inhibited, either using the dominant negative mastermind or siRNA, both IGF-BP1 and interleukin-11, which are both markers of destivalization, are completely suppressed. And you look at the conditioned media, you see the same thing, that in the presence of siRNA, under the destivalization response, you find that there is a decrease in interleukin-11 secretion from these cells, indicating that when Notch signaling is inhibited, you see a decrease in the amount of destitual response. We then went on to look at women with endometriosis. And if you now look at women with endometriosis and look at both the Notch receptors as well as at their ligands and downstream transcription factors that are regulated by Notch, all of them are suppressed in the eutopic endometrium of women with endometriosis. And then if you do the immunostaining, you can see again with Notch1, Notch4, and Hess, which is a transcriptional regulator of Notch, that they're completely attenuated in the endometrium of both women and baboons with disease compared to those animals without disease. And if you then take stromal cells from these animals or women with the disease and subject them to the destivalization response and look at the classic markers of destivalization, which is IGF-BP1 or prolactin, again, they are significantly downregulated, suggesting that the stromal cells from both baboons and women with endometriosis cannot undergo the destivalization response, similar to what we had shown if you just simply inhibited Notch signaling using siRNA. Using the same method, we then went ahead and did a microarray and tried to look to see what are the changes that are associated with the suppression of Notch signaling. And this is just a heat map showing you a large number of genes that are altered. But most importantly, what we found was if you looked at it globally and looked at the specific genes that were altered when Notch signaling was inhibited, the FOXO1 genes or the FOXO1 regulated genes were the ones that were most markedly affected. And that's really interesting because FOXO1, again, is an early inducer of the destivalization process. It works synergistically with Notch1 to allow for the development for destivalization to take place. And if you suppress Notch1, both in the context of the disease or using siRNA, we were able to show that all the FOXO1 regulated genes were significantly suppressed. So what we were essentially able to show in a summary here is that the destivalization trigger, which in this case was chorionic gonadotrophin, which alters the stromal fibroblasts and transforms it to a myofibroblast phenotype and increases Notch1, to allow for the changes in FOXO1 and all of the downstream genes that are important for the destivalization response, is synergistic with progesterone, and apoptosis is inhibited when the conditions are normal. However, in the context of endometriosis, what happens is that you see a suppression of Notch1, you see a decrease in FOXO1, which will prevent the downstream effects of these two important genes for the destivalization response, and that coupled with the progesterone unresponsiveness or the progesterone resistance that's part of this disease, there is a significant alteration that prevents the inability of these cells to undergo the destivalization response, which might be one of the mechanisms by which infertility is associated with this particular disease. So finally, what I'd like to do is just show you some work that we've been also trying to do in understanding the role of chorionic gonadotrophin as it relates to controlled ovarian hyperstimulation. And there have been a number of papers that have come out over the recent past suggesting either a positive or no response to the infusion of chorionic gonadotrophin into the uterus just prior to embryo transfer. There have been, and last year there was an extensive Cochrane review of all of these papers, and essentially what the Cochrane review concluded was that a large clinical trial still needs to be done, that there was no evidence to suggest that there was any effect on miscarriage or an alteration to the embryos, but yet that the literature, as I have shown you, that the number of papers was small, the number of patients in each of the studies was small, and so for us to be able to see whether HCG really has a role prior to embryo transfer, we need to do a few more definitive studies. But we were able to do one study, and these are using women that were embryo donors, and essentially they were undergoing controlled ovarian hyperstimulation, like as part of our IVF program, and these are young embryo donors, and we didn't interfere with any of the ovarian hyperstimulation, but they, with consent, we went in after oocyte retrieval, and the third day after oocyte retrieval, these women agreed to come back, and we infused the uterus with either 500 IU of chorionic gonadotrophin or conditioned media, and they came back 48 hours later for both a uterine lavage and an endometrial biopsy. And when we analyzed the endometrial biopsy, we first wanted to make sure that the chorionic gonadotrophin was having an effect, and we looked at the expression of phospho-ERK, because that's an indication of HCG responsiveness into the endometrium, and we saw that in the presence of HCG, you see a very nice localization of phospho-ERK in the luminal epithelial cells, and that was quantified using ETSCO. And there was also a remarkable effect on the expression of both the estrogen receptor alpha, and the progesterone receptor. So there was a direct response again in these women after controlled ovarian hyperstimulation in response to chorionic gonadotrophin for both the signaling mechanisms as well as the change in steroid receptors. And if you then looked at Notch1, which is one of our important targets, again, we see an enormous increase in Notch staining, not in Notch transcription, and that's normal. We've seen that before too, even with our BABOON model, that the transcript levels don't go up, but there is a change in the expression of the protein for Notch1, as well as the staining for complement 3, which is again another classic HCG-induced protein in the endometrium that came out in our microarray and was subsequently shown by the group in Chile, in Luigi Devota's group, that it is also regulated in the stromal cells by chorionic gonadotrophin. So what we were able to see in our model with controlled ovarian hyperstimulation in women with that were oocyte donors, so they were young women that were responding, that there was a synergistic effect with HCG and progesterone receptor. And again, the ERK pathway does alter progesterone receptor expression, as we've shown and others have shown, to regulate the progesterone receptor. And in response to this, what we find is that the change in both complement 3 and alpha smooth muscle actin, which are important for the immune priming for implantation, as well as stromal survival, because if you disrupt that cytoskeleton, these cells undergo apoptosis, and the upregulation of both Notch1 and estrogen, because there's close crosstalk between these two genes, to initiate again stromal cell survival and transformation of the destabilization process, suggesting perhaps in the context of trying to correct that endometrium, that the infusion of chorionic gonadotrophin before embryo transfer might be useful in resetting that endometrium. And our data that I didn't show you, we also saw that the dyssynchrony between the epithelial and stromal cells was corrected in the presence of chorionic gonadotrophin. So our human studies have shown that HCG does modulate various metabolic pathways within the endometrium and the decidua, that it reverses the apoptotic process that might be induced either because of dyssynchrony following ovarian hyperstimulation, or in situations where there are luteal phase defects and there's early effects to the progesterone, decrease in the progesterone, and perhaps that can be improved to increase CL support. That the intrauterine HCG prior to embryo transfer seems to counteract some of these effects, and that might be helpful in terms of correcting the endometrium, and that it does seem to have a clinical impact on endometrial priming and HCG prior to embryo transfer. But these are very small studies, and more studies are needed to obviously make sure that the similar responses that we see using our non-human primate model can be translated into the human situation. So what I've tried to show you is that if you look at the embryonic signaling, just like in other species, the primate, both the human and the non-human primate, does use embryonic signals in addition to maintaining the corpus luteum. It does use it significantly to allow for maintaining the endometrium, correcting the endometrium, sustaining it during those early events that are so critical for the implantation process. So with that, I'd like to thank you all very much and thank Milton for the invitation to come here and be part of this exciting meeting. And I acknowledge all the people that did the work. The people highlighted in white, Yalda, Afsha, and Praga Banerjee were graduate students that are now on their own, that did a lot of the initial work on the HCG, and all of the endometriosis work was done by Renvi Su when he was a postdoc in the lab. And then I've had really good collaborations with Bruce Lessie, whom we've done a lot of the signaling work with. Andrew Sharkey and Rob Sherwin at Cambridge helped us with a lot of the early microarray studies. And Lucio Mile did work with us with regards to all the NOT signaling, and Carlos's group with the ERA work that we also did on the HCG paper that I didn't get time to talk about. So thank you very much, and I'll be happy to answer any questions.